Okay, with that, I think we'll go ahead and get started. A few administrative details before I introduce our speaker this evening. So we encourage you all, if you have not already, to join ASM, both the branch here and nationally. It really does connect all of us with our peers, and there's some great opportunities to collaborate, talk with other folks that are interested in the same things that you are, and really ASM is the place for microbiology, including clinical microbiology, the topic of our lecture this evening. I'm going to launch a poll. I think we've done this the last 2 events. So what helps us as a branch? We know that our members have some diverse interests and come from a number of backgrounds, and getting a little bit of information about what you're interested in helps us plan these types of events. So our monthly meetings, our annual infection immunity forum, the annual symposium that's focused on clinical microbiology, and all of our outreach, scientific outreach and educational programs. So please fill out the poll if you have not done one already. If at the end, if you would like to put your questions in the chat, you can see the arrow here, you can place them in the chat, or if you would like to go to the participant tab and click on the blue hand to raise your hand, we can actually unmute you. So we welcome questions at the end of today's presentation. So with all of that out of the way, it is my absolute pleasure to introduce Dr. Bobby Pritt, who is Professor of Laboratory Medicine and Pathology and Chair of the Division of Clinical Microbiology at Mayo Clinic in Rochester, Minnesota. She serves as the Director of the Clinical Parasitology Laboratory at Mayo Clinic and is the Co-Director of Vector-Borne Diseases Laboratory Services. Dr. Pritt graduated from the University of Vermont College of Medicine, then completed a residency in anatomic and clinical pathology, followed by a fellowship in clinical microbiology at Mayo Clinic. She was hired on as staff at Mayo Clinic after fellowship, and then she obtained a master's degree in medical parasitology from the London School of Hygiene and Tropical Medicine and a diploma of tropical medicine from the Royal College of Physicians, London. Dr. Pritt shares her love of parasites through her weekly blog, Creepy, Dreadful, Wonderful Parasites, which you can actually check out at parasitewonders.blogspot.com. As a medical director within the Division of Clinical Microbiology at Mayo Clinic, she oversees the testing of specimens from around the world from patients suspected of having vector-borne disease. Mayo Clinic Laboratories is a high-volume global reference laboratory that performs more than 200,000 tests specifically for the detection of vector-borne pathogens. In North America, her laboratory receives specimens from all 50 states and Canada, with the majority originating from regions of high endemicity for tick-borne diseases. Through her work on vector-borne diseases, Dr. Pritt led the collaborative effort with the U.S. Centers of Disease Control and Prevention to describe and characterize 2 novel tick-borne pathogens, Borrelia mayonii and Ehrlichia murus occlurensis, that were discovered in her laboratory. Dr. Pritt has authored more than 195 publications in peer-reviewed journals, including recent publications in the New England Journal of Medicine and the Lancet Infectious Diseases, in which she and her team describe Borrelia mayonii and Ehrlichia murus occlurensis as human pathogens. Today, she will be discussing her work in new tick-borne pathogen discovery with her talk entitled What's Bugging You? Ticks, Teamwork, and Emerging Pathogens. Dr. Pritt, I'm thrilled to have you here, so please take it away.
Well, thank you, Kyle. It is my absolute pleasure to be here from across the ocean. I'm in Portugal, and we were just saying how wonderful it is that I can actually join you virtually, even though I'd love to be there in person, just to have this opportunity, though, to talk about one of my favorite subjects, which is tick-borne diseases. So my topic for today, my title is What's Bugging You?, focusing on tick-borne diseases in the United States with a particular emphasis in endemic areas, including where all of you live. So it's a mixed blessing. It's wonderful science, but not so wonderful when you have to worry about these little critters. So we'll talk about the tick-borne pathogens and what's in your area and what you need to worry about. So with that, I thought I would start by setting the stage, or in this case, the forest, on what are tick-borne diseases, why are they relevant. So let's start by talking about ticks. So tick basics, they are arachnids. They are not insects, so they are actually more closely related to mites and spiders. They have 8 legs as adults, where insects have 6 legs. So that's one of my pet peeves, is when the media always calls ticks the insects that transmit Lyme disease, because that's incorrect. They're further divided into hard and soft ticks based on their body type. The hard ticks are in the Ixotidae family. The soft ticks are in the Argacidae. I'm really going to focus on the hard tick transmitted diseases, which by far are the #1 cause of tick-borne pathogens for humans. And regardless of soft or hard ticks, they all have these 4 life cycle stages. So they start off as an egg, they're laid as an egg from their mom, they hatch into a larva, then they molt into a nymph, and then they molt again into an adult. And each step requires a blood meal. So they are obligate blood-sucking ectoparasites. They need your blood in order to go from one stage of their life to the next. So let's think about their life cycle here. This I'm using as the classic life cycle. This is specifically for Ixodes scapularis. And we're going to start here with the mom tick. Pregnant mom, she's gravid, she's full of eggs, and she gives all of her eggs here. She lays them up near her head, which is an interesting place to lay your eggs. Now, some of these pathogens have transovarial transmission. So that means that some of these are actually born with pathogens already. That does not apply to Lyme disease, thankfully. Lyme disease is not transmitted from mom to egg, but some of the viruses and also Borrelia miyamotoi is. So once they hatch, that is right here with the eggs, they hatch into the larval stage. They're very small, about the size of a period at the end of a sentence. Very teeny, and they feed on teeny hosts like birds and small rodents. So let's just take a look at them. Here's some lovely little ticks that we hatched in my lab for fun. We don't do this for a clinical basis, but my technologist just thought it was really fun. So we did this. If you want to do it, it takes about 6 weeks, just in case. We did kill them all afterwards because they are blood-sucking ectoparasites. So here's baby's first steps, and they're going to then go out and they're going to exhibit this behavior called questing, which is when they go to the top of a high surface nearby, which for these little guys is just the edge of a leaf or a little blade of grass, and they extend their legs so that they can catch on to the first animal that walks by, which in this case is probably a little mouse, like the white-footed mouse, Peromyscus leucopus, or a little bird. Now, that little mouse or that little bird may be infected, and if that's the case, when they took that blood meal, they are then going to be infected. They will overwinter, and when they overwinter, they will come out as nymphs, and these nymphs are now infectious because they picked up that infection. If they weren't already born with it, they got it when they took their first blood meal. These nymphs are about the size of a poppy seed, but yet they're infected and they're now going after the larger hosts. And this is where this is the most dangerous stage for humans because they're so small, they're hard to see, but early in the spring, they're out and they're biting and they're potentially carrying dangerous pathogens that can pass on to you. They take their blood meal. If they weren't infected the first time, they may become infected the second time when they take their second meal. And then again, they will molt and now turn into adults. And the adults, again, can overwinter, even in our bitter, cold, miserable winters in the upper Northeast and the upper Midwest. And then the mother tick will come out and lay her egg. So it's usually a 2-year cycle for the Ixodes scapularis or deer tick. So here she is. This is a female deer tick extending her legs, exhibiting that questing behavior. So what do we know about tick-borne pathogens? There's quite a number of them. Starting from the early 1900s, we had 7 pathogens. You may be surprised to know that Rickettsia rickettsii, the agent of Rocky Mountain spotted fever, was one of the first that we detected. Tularensis, Borrelia turcati, some of the relapsing fever Borrelia, they were actually discovered fairly early on. So that's in the first 60 years from the turn of the century. And the next 60 years brought 12 pathogens. Now let's just kind of hone in on this period just within the past couple of decades and look at all of the pathogens, you can see it picking up pace, that we're detecting more and more pathogens. And this is where we see Ehrlichia muricoclorensis and Borrelia mayonii. I'll tell you that story in a little bit. Some of the new viruses, and then possibly some new things that we've been working on with the CDC, where we've detected some potentially new pathogens. So there are at least 9 bacterial diseases caused by 14 pathogens that are tick transmitted. There are 3 viral diseases and then 1 parasitic disease due to several species of Babesia. So if we put all of the vector-borne diseases into a big pie, well, Lyme disease makes up the majority. Lyme disease is the #1 vector-borne disease in North America, #1 tick-borne disease. The mosquito-borne diseases really make up a tiny slice of the pie. And then the remaining piece here are our other tick-borne diseases. So let's talk about those ticks and the pathogens that they carry. We're going to set the stage as a national stage. And the bad news is that, hold on, I'm just trying to close this. Okay, the bad news is that we have ticks throughout the United States. There's different distributions of different types of ticks, but their distribution, every state has at least one different type of tick. So Brachycephalus sanguinis, the brown dog tick, is in every state in the United States. Every one that is colored in orange is its distribution. This tick usually doesn't transmit pathogens, but it can transmit Rickettsia rickettsiae, which causes Rocky Mountain spotted fever, which is a potentially serious fatal infection. Then in your neck of the woods as well as ours, there's the wood tick, or sometimes called the American dog tick. So that's really the eastern part of the country. There's a different one over here, Dermacenter. Well, there's a different one right here, and then this is all Dermacenter variabilis. Now, this transmits a number of pathogens, Rickettsia rickettsiae, Francisella tularensis. Then we have this little tick, Amblyomma maculatum, down in the southern area, just dipping up or just going up into Pennsylvania. That transmits Rickettsia parkeri, rickettsiosis. This one you've probably heard of, the Lone Star tick, Amblyomma americanum, all throughout Pennsylvania, the Northeast, the Southeast. This transmits a number of important pathogens, Francisella tularensis, the Ehrlichia species, Ehrlichia chaffiensis and Ehrlichia ewingii, Stari, which probably isn't infectious, but no one knows exactly what causes it. Possibly or probably Heartland virus and Bourbon virus. And then last but not least, it is associated with meat allergy, and some people actually think that's worse than getting ehrlichiosis. It's not. Ehrlichiosis is potentially serious, but being allergic to meat and not being able to eat it for a decade could be quite serious in some people's mind. And then last but not least, probably the biggest culprit of all of the ticks is the black-legged tick, formerly known as the deer tick, Ixodes scapularis. And that's because this is the tick that transmits the organisms that cause Lyme disease. So we have Borrelia burgdorferi, and one we'll be talking about shortly, Borrelia mayonii, anaplasmosis caused by Anaplasma phagocytophilum, transmits Babesia species, Powassan virus, and Borrelia miyamotoi. The other one we're going to talk about in a little bit is Ehrlichia muricoclorensis. The good news for those of you living on the East Coast is Borrelia mayonii and Ehrlichia muricoclorensis seem to only be localized in the upper Midwest. So 2 less pathogens for you to worry about, but certainly this tick is found in the Northeast, along the East Coast, and definitely is a major vector of important pathogens. So now I get to go into the road to pathogen discovery. Even though the 2 pathogens we discovered are only found in the upper Midwest, I think it's an interesting story, and it goes to tell a little bit about what is involved when you discover a new pathogen, how you work it up, how you give it a name. It's probably more complicated than you would think, and it's an interesting process. So I was very honored and fortunate to go through this process. So let me tell you the story, the way it unfolded for me. So we're first going to start how the story began. It was in June of 2009 in Wisconsin in the Mayo Clinic health system. And we have this area called Eau Claire, that's the city, and we have a Mayo Clinic site there. And they're pretty advanced. It's a pretty large lab. They're not as big as our Mayo Clinic Rochester lab, but they do a lot of PCR testing. So for a small community hospital, they're fairly sophisticated. So they were running a PCR test for Anaplasma phagocytophilum. And it was these two astute technologists that noticed that something was off. They were finding an atypical result, and it was repeatable. Now, why were they performing a test for anaplasmosis? Well, there's a lot of it up in the upper Midwest, where they were in Wisconsin and where I am in Minnesota, and also along the East Coast. They also have a lot of Lyme disease, and they have a lot of babesiosis. So they were performing tests for all of these pathogens. And these are all transmitted by the same tick, that black-legged tick. So they were using this assay that was designed by Dr. Robin Battelle back in 2005, and this is in the public domain. It's published, so numerous people use this assay, and that becomes important in the story. It's nice when you have more than one person using the same assay, because then they can find similar things that you can find, or different things for that matter. So this assay targets a region that is conserved amongst the Anaplasma and Ehrlichia species. That's the GRO-EL heat shock operon gene. And then using something called melting temperature analysis, after you've amplified your DNA, you can differentiate Ehrlichia chaffiensis, Ehrlichia or Ewingii chaffiensis, and Anaplasma phagocytophilum. Now, they were running this just for the Anaplasma. They really could care less about these two organisms. But that becomes relevant because what they detected was an unusual peak right between the two Ehrlichia species. And this was not expected. It was reproducible, and its location made them think that they might actually have an Ehrlichia species. Well, so here it is. There's only two base pair changes between Ewingii and chaffiensis. So the fact that they had this peak made them think, okay, well, the primers were able to bind and amplify this portion of DNA. And then the melting temperature peak, that has to do with the probes, and the probes annealed and produced fluorescence. And during this melting temperature peak, you had a peak right between the two Ehrlichia species. So because the primers and the probes had both bound, they knew it was in the Ehrlichia family. They just didn't know what it was exactly. So let's talk about Ehrlichia species. If I was in there in person, I'd ask for a show of hands of how many people have heard of Ehrlichia. Some of you may have. It is in Pennsylvania. So Ehrlichia species are closely related to Anaplasma phagocytophilum, bacterial tick-borne pathogen. And in this case, it is an obligate intracellular bacterium. And I see that I have a hand raised here too. So hold on a second. Let me. Oh, yeah. Looks like Patricia. Do you want to ask a question?
Not at this time. Thank you, Doctor.
Oh, okay, no problem. Oh, was that just my show of hands? Thank you. That's awesome. I could ask for everyone to do a show of hands, but I won't do that now. If you've heard of Ehrlichia, this is related to Anaplasma. It's obligate intracellular. This is not a bacteria that can grow on a blood agar culture plate. It's only going to be found in certain types of media because it has to grow inside of cells. So it's transmitted by the Lone Star tick, which is interesting because that's not found in any extent in Wisconsin. So this was not expected. They infect white blood cells. They cause this febrile illness, rarely can have complications involving multiple organ systems, and very rarely can cause death. So this can be a serious infection. But this was not thought to be in Wisconsin or Minnesota. So our poor techs were trying to figure out why they had a patient that was testing positive for something that looked like Ehrlichia when they were running a test that really was, in their mind, just designed to detect Anaplasma. But what do we know about this patient? Here we have patient number 1, and notice that this is the patient who has this atypical melting temperature peak between the two Ehrlichia species that were known at the time. So this patient was a 54-year-old man from Wisconsin. He did have ticks on him at his cabin. Now, you should know that most people that live in Wisconsin have a cabin or a family member who has a cabin. They like to spend time in the woods, and ticks are a part of life. So he didn't remember any bites, but he certainly knew he was exposed to ticks. He had a sudden onset of fever, fatigue, muscle pains, and headache. So this looked like a febrile illness. Here it was in June. His laboratory values looked like he had a tick-borne illness. He had low platelets, a lymphopenia, slightly elevated liver function tests. These are all signs of a tick-borne illness, but all of the tests for the known things were negative. All that was positive was this unexpected finding. So this was starting to make it look like he actually had something that was attributed to whatever this PCR was indicating. He also got better after receiving doxycycline, which is the drug that we use for tick-borne pathogens, for the bacterial pathogens. So this is interesting. You know, was this just a weird fluke that we detected this one patient? Well, we did report it, so we reached out to, first we had them send the isolate to us here at Mayo Clinic in Rochester, so that's southern Minnesota. We worked it up a little further. We sequenced the GRO-EL heat shock operon gene, and here we had the Wisconsin specimen, not closely related to Ehrlichia chaffiensis or Ewingii, but instead clustering with Ehrlichia murus. And as the name implies, this is a pathogen found in mice, but never found in North America before. It was only found in Japan and Russia. So what is this pathogen that was never found in humans before and never found on this continent doing in Wisconsin? So we contacted the folks at the Wisconsin State Health Lab, and that was Zip Johnson, and they said, let's send it to the CDC. So we sent the specimen to the CDC, and sure enough, they worked it up, this time sequencing a different gene, the 16S ribosomal rRNA gene. And here is our specimen, the Wisconsin species, again, with a different gene, showing that it's not closely grouping with Ewingii and chaffiensis, but instead grouping most closely with Ehrlichia murus. So we started calling this the Ehrlichia murus-like agent because we had nothing better to call it at that time. This is where it could have just stopped. In a discovery process, what would we have done? This would have been a case report, and that would have been the end of the story. But then patient number 2 happened the very next month. And it was also detected by this atypical melting peak that looked identical to patient number 1. Now, this patient was younger, 23-year-old male from Wisconsin, so another patient from Wisconsin. But he had received bilateral lung transplantation for cystic fibrosis. So he was an immunocompromised man. And in addition to having fever and fatigue, he also had a left lung infiltrate and a pleural effusion. This was worrisome enough to hospitalize him. And they did so at my hospital at Mayo Clinic in Minnesota. So he was hospitalized for 3 days. That was enough time for me to talk to him, get consent to do another blood draw, and obtain blood for culture and additional testing. So now I have this blood and I want to culture it. But remember, I told you it's an obligate intracellular bacterium. I can't just streak it on a blood agar. It only grows within cells, very much like a viral cell culture. But I don't do viral cell culture. So I had to reach out to someone who did. So I, this time, talked to Dave Neitzel at the Minnesota Department of Health, since this patient was getting reported to Minnesota since he was hospitalized in Minnesota. And he said, well, we don't culture it at our Department of Health, but you should reach out to Dr. Uli Munderloh at the University of Minnesota. So I cold called her and said, hi, Dr. Munderloh, you don't know me, but I have the specimen and is there any chance that you would culture it for us? So I would say one of the first, besides the fact of just be curious, and when you see things that are unexpected, you should follow them up. My second lesson would be when you're working up something like this, don't be afraid to pick up the phone and call people. In general, they're going to be like-minded fellow scientists, and they're going to be just as curious and want to participate as well. So I said, well, I have this blood. Can I come up and give it to you? So I drove up to the Twin Cities, which is about an hour and a half away, and she met me at her door, and we went over to her lab, and she inoculated into several cell lines. And then she said, okay, now we have to wait 6 weeks because it can take that long. So we waited and waited and waited, and I kind of forgot that it was just still cooking away. And then I get this excited phone call out of the blue. And she said, something is growing and we can see it in the cell lines. And she sent me these pictures of these Ehrlichia morelli, which is the name for the clusters of bacteria and how they grow inside cells. So let me show you. This here is a single cell. The light purple object is the nucleus, and these are the clusters of bacteria growing in the cytoplasm. Here's a different one with the nucleus, and then this has multiple smaller clusters. So these are just two different cell lines. So this was growing, and if we hadn't been able to grow this in culture, we would have had a much harder time publishing it and confirming that it was a novel organism. So reaching out to people is important. Don't try to do it all yourself unless you can, I suppose, but you know, sometimes it's better to have a group of people and have different minds with different viewpoints. She did her own sequencing and also found that this was an Ehrlichia mirus-like organism. It was definitely not any of the known species. So we were pretty excited at this point. We reached out to the CDC again, and we really got their attention because then they deployed the Epidemic Intelligence Service. So these two officers came to my laboratory. I gave them a tour. I explained how our PCR worked. They collected specimens. They interviewed patients. So now it was a formal EIS investigation, which was pretty exciting that summer. Now, meanwhile, I'm asking myself, well, if this is a novel pathogen that is related to tick-borne pathogens, it's probably transmitted by a tick vector. But what tick is it? Because in Wisconsin, we have Dermacentor and we have the black-legged tick Ixodes scapularis, but we do not have the Lone Star tick, which is the known vector of Ehrlichia species. So it must be some other tick. Which one is it? So I, again, don't usually go out and collect ticks in my free time, but I learned how to do that. I reached out to Zip Johnson, and she said, well, you should talk to Dr. Susan Paskowitz and Dr. Zia Lee from the University of Wisconsin, because they know all about going out into the field and collecting ticks. So I teamed up with them. I had my development technologist, Lynn Sloan, who was such a good sport. She came with me. And we went out, here we are on my property, actually in Wisconsin, and we use these big white flags to drag it over vegetation. The ticks will grab on because they think it's their next blood meal. They think it's a little animal. They grab onto it, and then you can examine the white sheet. Usually it's made of something like flannel, which is stickier for the ticks. You examine it really carefully, you take the ticks off, you put them into little vials, and then you clean it off with a little pet lint roller, and then you drag it again. So we collected all the ticks, we brought them back to the lab. We did just a very crude mincing of the ticks. We digested them in proteinase K, and then we tested them with the same PCR that we were using for patient whole blood. We also had some archived tick specimens that Dr. Paskowitz gave us, so we tested those as well. So we're testing a few hundred ticks, looking to see if we can detect this new Ehrlichia mirus-like organism. Well, then I get this phone call out of the blue from Dr. Ellen Stromdale. She was working at the Department of Defense. This is Dr. Stromdale right here, teaching tick safety to some park rangers. And she ran the tick testing program for the Department of Defense through the U.S. Army. In this program, she would take ticks that were identified by any military personnel on their body, also any civilians that happen to be on military bases. And every dot on this map was a military base from where she collected ticks. And the white ones were the primary centers. So she got a lot of ticks from people there, including one in Minnesota and one in Wisconsin, and then some smaller tick centers. And she would take these ticks and she would test them for all sorts of different pathogens. And it just so happened she was using our Anaplasma Ehrlichia PCR. Remember this one? And she was seeing that same atypical melting temperature peak that we were seeing, but only from ticks in Minnesota and Wisconsin. She did not see this in any of the other ticks collected from any of these states, from any of these military bases. So again, that's the nice thing of using an assay that's in the public domain that other people could bring live. So we decided to pull our data together And our results showed that this new Ehrlichia mirus-like agent, or EML agent, was only found in black-legged ticks, Ixodes scapularis, not the Lone Star ticks, and only specifically black-legged ticks from Minnesota and Wisconsin. It wasn't found in any Ixodes scapularis from any other state, more than 2,500 ticks, so fairly decent sample size. Also, it was not found in over 6,500 Amblyomma americanum Lone Star ticks from all different states in the South and Southeast. So it didn't look like that was our tick vector. Very interesting. So all in all, we had a lot of findings, clinical findings. It ended up being 4 patients, tick findings, and we published them all, and this was quite the endeavor. You could see all of the names on this publication, all of the different institutions, the Minnesota Department of Health, the CDC, the Wisconsin State Health Lab, 2 different universities, and then Mayo Clinic. So incredible experience. And we followed it up with a second publication to talk about what we were seeing with this pathogen from 2007 to 2013. At that time, out of over 75,000 patient whole blood specimens we had tested from all 50 US states, we had 69 positive. So 0.1%, obviously very low, but this is from all of the specimens from all 50 states. And these are positive with that same atypical melting temperature peak that we knew now was indicative of Ehrlichia mirus-like agent. And it nicely broke down that there were 33 patients from Minnesota, 31 from Wisconsin, and then we had a few scattered from surrounding areas, but they had all had recent travel to Minnesota or Wisconsin. So there's something special about our ticks that they have this Ehrlichia mirus pathogen and the rest of the country does not. So it was not found in any other state, not in ticks and not in human blood specimens. And out of 75,000 that we tested from all 50 states, that was a pretty decent sample size for us to draw that conclusion. And that is despite the fact that we test specimens from all over. You can see every blue dot on this map is a client that sends specimens to my laboratory for tick-borne disease testing. And there's a lot of specimens in the upper Northeast, including some in Pennsylvania. And none of those human blood samples, tens of thousands, were positive for Ehrlichia mirus. It was just the ones from the upper Midwest. Now, these patients were of male predominance, which is pretty common for ehrlichiosis and anaplasmosis. Median age was 62, also fairly common. More than a quarter were immunocompromised, which we thought was quite interesting. That may have a bit of a reporting bias, though, because those patients may have been more ill and therefore more likely to be tested by RT-PCR. So hard to know. Remember, this is not us doing active surveillance, going out and testing everyone. These are just people that had their blood tested in my laboratory. And we found that the clinical and lab manifestations are pretty much what you see with ehrlichiosis. So they have fever, it's a febrile illness with malaise and fatigue, headache, rarely rash, thrombocytopenia, lymphopenia, elevated LSTs. And this is not a benign disease. Almost 30% were hospitalized, but fortunately there have not been any deaths to this day. And we continue to track the patients. We've now had several hundred. Still no deaths, fortunately, so relatively rare disease, but only found in the upper Midwest. So what are we going to call this? We can't keep calling it the Ehrlichia mirus-like agent. So we had to do a number of studies so that we could finally give this a name. It was accepted as a species of Ehrlichia mirus, but it was a subspecies that was unique to North America. So we named it Ehrlichia mirus oaklerensis after Eau Claire, Wisconsin, where we first identified it. So that was published. So now we could just call it E. mirus for short. If you're talking about North America, it's Ehrlichia mirus oaklerensis. And so that was a whirlwind over a couple of years in describing a new organism. And if you're curious in the end and we have time, I can tell you more about what goes into coming up with a suitable name for an organism. So then after we get this published in 2011, it is just the following year or 2012. So the following year, the summer of 2013, we have almost an identical story, but with a different PCR and a different clinical presentation. But I thought, wow, this can't really be happening again, can it? So we had now an atypical Borrelia Lyme disease PCR result. So let's talk about this patient. This was a patient that lived in Northwest Wisconsin, but he had spent some time just the week prior in Spooner, Wisconsin. So still in that upper Midwest area. And he's only 10 years old. He presented with fever, headache, neck pain, myalgia, nausea, vomiting, a diffuse rash. It did not look like your typical bullseye rash of Lyme disease. And he had profound somnolence. And you may say, well, what does that mean? He was sleepy? He was so sleepy. His parents had a hard time rousing him, like they had to shake him so that he could actually respond to them. So very worrisome, as you can imagine. He was so sick that they actually had him hospitalized at Mayo Clinic in Rochester, Minnesota. And his parents took this picture and shared it with us with their consent. And this is the rash he had. And they described it as more of like a Dalmatian. It was spotted all over his body. None of them looked like a bullseye rash. And they threw the kitchen sink at him. They really did not know what he had. They ran every single test they could on him that seemed relevant. And the only one that was positive was this, our Lyme disease PCR. We don't recommend Lyme disease PCR on whole blood. It's usually not considered a very good way to diagnose Lyme disease. All of you are probably thinking, that's right, it's serology we use for Lyme disease. And I was actually thinking of discontinuing this test because I had inherited it and I thought, well, this is not very useful and people are probably using it and thinking they're getting a good test. It's not that the test is bad, it's just that usually you don't detect Borrelia burgdorferi in whole blood. But this is what we saw, an atypical melting temperature peak right here in between the expected melting temperature peak for Borrelia burgdorferi and Borrelia afzelii and grinnii. These are the two species in Europe that cause Lyme disease. This is the one in the United States that causes Lyme disease. And we had something smack dab in the middle. This assay is designed to detect and differentiate members of the Borrelia burgdorferi sensu lato genocomplex. So basically all the different Borrelia species that are responsible for Lyme disease, and then differentiate them using melting temperature analysis. But we never expected to see something right here in between the two. So what do we know about this patient? Well, he was hospitalized for 4 days, treated with antibiotics. Fortunately, he made a complete recovery. Oh, I will note, it was interesting. Not only did he have this spotted rash all over his body, it also involved the palms and the soles. Not a common distribution for any type of rash. It's only a few organisms that do that. So as we were working this case up, we started having more cases. We had another PCR positive in whole blood from an 11-year-old male in Wisconsin. So then I started thinking, well, is it possible that we had some of these and they were just signed out as atypical? So sure enough, we looked back and a retrospective review from the prior year showed a 65-year-old male who had that same result. We didn't have the blood anymore on that patient, but we still had this result. And he had exposure to ticks in Minnesota. And then, interestingly, we had a synovial fluid. So it wasn't just whole blood, but we had a synovial fluid that also had the same PCR result from a 21-year-old woman in Wisconsin. So we now had a series of patients. We did sequencing on them. We started with the gene we amplified for our PCR, which happened to be this plasminogen binding protein gene, and found that these organisms were clustering by themselves, very different than all the other known Borrelia species. And it was about 89 to 95% similar. So we thought we were actually dealing with something completely new this time. So now, if we have any students in the audience, you could ask yourself, now what? What would you do if you were in this situation? Well, thankfully, we had all these great contacts. So I just picked up the phone and reached out to our Minnesota Department of Health and the Wisconsin and University of Wisconsin, University of Minnesota. So that's another lesson for any of the students in the audience is make those contacts and then you just have like friends for life. You can pick up the phone and call. And so this time it was slightly different, but very similar players. I reached out to Dave Neitzel from the Minnesota Department of Health, Zip Johnson from the Wisconsin State Health Lab, and this time it was the CDC branch in Fort Collins in Colorado. It was not the Atlanta, Georgia branch. So they're a little siloed. All the Lyme disease is done in Fort Collins. So Dr. Mead and Dr. Peterson were wonderful to work with. They said, send us some blood. We want to try to culture this organism, and we want to do some sequencing. So we had blood from 2 patients. It was a few weeks old, but they thought it would still be worthwhile sending, so we did. And they were able to culture it and sequence it. We also performed that at Mayo Clinic. And that was all, again, to my wonderful development technologist who did all this work, because if any of you have ever tried to grow up Borrelia spirochetes, it is not an easy task. You need to use a special liquid-based media. Again, this is not something that grows on a blood agar. So in case you're wondering, this is what a little spirochete looks like. We actually saw them in peripheral blood diluted 1 to 10, and here comes another one. Up it goes and back down again. So we'll let that loop and you can see it go. This is very uncommon that you would actually see spirochetes in the peripheral blood smear in a patient that has symptoms consistent with Lyme disease. And you can also see it right here. So we have a spirochete on peripheral blood. We also were able to grow it in culture. And here are Borrelia spirochetes in culture. That corkscrew motility of a spirochete, very classic. So again, this isn't something we could have done by ourselves. This was truly a team-based approach. So we decided to really try to figure out how this organism was related to others. So we did this phylogenetic analysis of 8 different housekeeping genes. And that showed that this was definitely not Borrelia burgdorferi. It clustered by itself here. We used the outgrouping as the relapsing fever Borrelia. It was completely unrelated to those. And then we did a multi-locus sequence analysis. And with these 8 genes, we were able to show that this was definitely a novel Borrelia burgdorferi sensu lato genospecies. It wasn't Borrelia burgdorferi. It wasn't any other known Borrelia species. So this one, again, if we have time, you could ask me about this if you're interested. We decided to call this one after the Mayo Clinic, the Mayo brothers that found at Mayo Clinic. So we called it Borrelia mayonii. So the key findings with this organism is that, well, first of all, interestingly, patients don't usually have that classic bullseye rash, but you will detect it with the classic Borrelia burgdorferi serology tests. It's only found in the Midwest. So Dr. Rodino is probably also grateful that he's moved out of the upper Midwest and doesn't have to deal with these 2 pathogens that we have up here. It has a propensity, or may, for causing neurologic manifestations. It's seen in peripheral blood, and it has high loads of bacteremia. And so you could actually detect it by PCR. In fact, that's probably the preferred method for detecting it. So there's many of the same players, some new ones as well. We published this and described the findings, and we now have this second pathogen. So you can put all of these diseases on this map, and I really like this map. It's pretty impressive. Look at all the tick-borne diseases in the upper Northeast, in the upper Midwest, scattered along the South and Southeast as well. Pennsylvania's in there somewhere. I think it's kind of obliterated. I could hardly see it. With all those tick-borne pathogens, and primarily you're dealing with Lyme disease there, but some babesiosis and others as well. So this was all well and good that we were able to just, by luck, serendipity, a little bit of, good assay design, detect these 2 new pathogens. But how can we go out and do this in a more prospective, deliberate manner? That's where this new collaboration called TickNet comes in. It actually isn't that new anymore. It was founded in 2007. It's a collaboration funded by CDC, provides funding to state and local health departments. And so we partnered with the Minnesota Department of Health and CDC, and we've been giving them residual waste samples, residual patient samples that would have been discarded. And of course, we're doing this all with consent and IRB approval. So we have given Mayo Clinic over 30,000 whole blood specimens and some CSF and synovial fluid as well, all submitted to my lab from patients that were suspected of having a tick-borne pathogen. And the CDC has been performing 16S, and they published this. So they did the 16S sequencing using next generation sequencing. We've done one publication with a smaller number of samples, and we're going to do the whole 30,000 soon. And already using this assay based on the variable regions 1 and 2 of the ribosomal RNA gene of over 13,000 specimens, we had a small number that were positive for tick-borne pathogens. And of these, we have at least 3 new pathogens, Borrelia johnsonii, and then a novel Ehrlichia species, or a novel Anaplasma and a novel Rickettsia species. So this is probably the way to do it, is being more deliberate, going out and actually looking for things. And there's probably more out there. The 16S, for those of you who know, that gene only is found in bacteria. So this is not going to detect our viruses, and there's probably plenty of those as well. There was a whole number of other organisms we detected too that probably weren't even suspected in these patients. It really goes to show the value of this kind of broad range targeted metagenomic approach. And then Dr. Rodino and I brought in an assay at Mayo Clinic, very similar, but using a V1 to V3 region of the ribosomal RNA gene. So that will hopefully be going live. This is probably too small to read, but essentially we have this tick-borne algorithm, and where I picture this entering our tick-borne disease testing algorithm would be at this point, after you've done your normal tick-borne testing, if the patient still if the patient's negative and you're not sure what's going on, at that point, we would go ahead and add in the tick-borne 16S ribosomal RNA testing for just a broader bacterial coverage. Eventually, my goal is to do rapid sequencing, maybe using something like the Oxford Nanopore MinION or GridION, and actually produce a result within 4 to 6 hours. I think that's the future, really, are these either targeted or shotgun metagenomic approaches. So this is my last slide. I'm going to leave you with some outstanding questions. These are questions that I have, at least. Well, first of all, why do some organisms have defined geographic ranges? Why do these 2 new tick-borne pathogens that we described, why are they only found in the upper Midwest? That's a little odd, isn't it? It's the same black-legged tick that's found all throughout the Northeast. So is it there and we're just not finding it? We've tested hundreds of thousands of specimens and it doesn't seem to be there. So more to come on that. Also, how do we do a better job at active surveillance and actively going out and finding what's out there? Partnerships with the CDC are useful. They're capable of testing 30,000 clinical specimens at a go with a next-gen sequencing assay, but are there more affordable, more scalable ways that clinical labs can do this testing? And then what else is out there? It really makes me wonder. I think the viruses are going to be the next big area and opportunity for discovery. So with that, I will end, and I believe we do have a few minutes for questions, so I would be happy to take any, and thank you all for your attention. And I see that Patricia has her hand up. Did you have any questions, Patricia, or was that from before? So maybe not. Does anyone else have any questions?
Thanks, Bobby. That was absolutely fantastic. We do have a few questions in the chat, and I think Jason's going to hop on here and help me run the Q&A session too. But the first one from Cynthia Flynn, did any of your immunosuppressed patients get these novel tick-borne pathogens by blood transfusion?
You know, that's a great question, Cynthia. So to this point, or at this time, we don't have any evidence of these being transmitted by blood transfusion, but we certainly know that other tick-borne pathogens like Ehrlichia chafiensis, Ehrlichia ewingii, anaplasma have been shown to be transmitted by blood transfusion. So I think it's possible. It's just we don't have any evidence at this time. And our patients that were immunocompromised weren't necessarily getting transfusions, but they were being treated for malignancies. So it could just be that maybe they were more susceptible to having severe disease and then got tested by PCR. But I think it's possible. I think that it's something that if you live in a highly endemic area, it may be something that we eventually screen our blood donors for. And let's see, I also see a question from Edward Shaw. So since serology is typically used to diagnose and the Mayonii patients zero cross-react, do you think there are cases being misdiagnosed as Bergdorferi that are actually Mayonii? Absolutely. So Edward, excellent question. We are, I'm sure, underdiagnosing actual cases of Borrelia mayonii because the serology cross-reacts, so we're probably detecting them all. As far as we can tell, the patients get better with doxycycline. So as long as they're treated, they get better. They probably never know they have Borrelia mayonii. The differences, though, that I find interesting are a potential diagnostic pitfall because in our hands, in our cases, the patients that we saw with Borrelia mayonii did not have typical rashes. And for those of you who deal with Lyme disease, you know that that rash is really important in guiding therapy. Early on, if you have a bullseye rash in the right clinical setting and epidemiologic setting, you just assume that patient has Lyme disease and you treat them. And if they have spots all over their body that looks like a Dalmatian, as the little boy's parents had called him, you may not treat that patient like they have Lyme disease. And so I do worry about potential misdiagnoses. The other interesting thing is that with the really high spirochetemia, the PCR is actually a really good test for Borrelia mayonii. But how do we encourage PCR for definitive diagnosis without overusing the PCR for other for Borrelia burgdorferi? That's a hard question to answer. I would say that for the most part, we're not using the PCR, and so we're probably just calling everything Borrelia burgdorferi.
I'm gonna.
Now let's see. Yeah, go ahead. I see another question in the chat, but I also see that Arthur has his hand raised.
Yeah.
Yes, I thought it was a fantastic presentation. I just wondered if you could comment about, tick distribution, because I know like Amblyomma americanum, it seems like that distribution has slowly been moving north.
Mm-hmm.
So, I mean, there's a role for actual changes in distribution of the ticks over time. So what you might not see something now, but you could see it in the future, depending on that distribution.
Yeah, Arthur, that's an excellent point. And that Lone Star Amblyomma americanum is a very aggressive biter. It transmits Ehrlichia chafiensis and Ehrlichia ewingii, as well as stari and meat allergy and everything else. It's not a tick that you want to have. And right now, it is not found in any appreciable numbers in Minnesota. But every year, the Minnesota Department of Health goes out and they find ticks, probably brought up by migrating birds. And with global warming, the ticks are probably more likely to actually survive. If our winters get milder, the Amblyomma americanum usually doesn't like severe winter. With our milder winters, they're probably going to be able to overwinter, and the ticks are going to move north. We're also seeing increasing tick ranges with Ixodes scapularis. We're seeing tremendous migration of ticks from the Northeast, expanding from the Midwest, and probably eventually there's going to be just this continuous swath that goes from the Northeast right into the Midwest. And then you have to worry about the pathogens like Borrelia mayonii spreading east with all of the other Ixodes scapularis.
Yeah, thank you very much.
You're welcome.
Yeah, and there are some couple other comments and questions in the chat that I think you can see. One from Matt Pettengill, who highlights that this is Lab Professionals Week. So
yes,
congratulations to everyone.
Yeah, and so let me just say congratulations, happy Lab Week. And if you want a good laugh, I participated in a microbiology Pictionary. And it's being posted. It was already posted on YouTube, I think today or maybe yesterday, and you can find it on Twitter as well. So I team up with 3 other excellent microbiologists, and it's pretty fun to watch. If you want to see me trying to draw with my mouse on a Zoom whiteboard in Microbiology Pictionary, it's kind of fun. We did it just for Lab Week.
I think you just answered this one with one of your last answer, but Toby asked if any of the pathogens had any resistance, or were they all susceptible to doxycycline?
Yeah, so far it does look like they're all susceptible to your standard treatments, which would mostly be doxycycline, a little bit of ceftriaxone for more severe CNS manifestations of Lyme disease. So that's good news. I think that as long as we can still detect these organisms and we can still treat them, then it's really more just, for the science and epidemiology of knowing what they are specifically, but at least it's not harming patient care if we don't know exactly what someone has. Now, the viruses are going to be a different story, though. So if any of you want to do studies in an emerging area, I think looking at viral pathogens that are transmitted by ticks is going to be a really interesting area.
Great. One more from Dieter. He asked, any small mammal potential reservoirs that are unique to the 2 states, Minnesota and Wisconsin, that could be the link to why this is limited to that geographic region?
So that's an excellent question, Dieter. And the answer is no. It's the same white-footed mouse, Paramyscus leucobus, one of the red squirrels, and chipmunks for the 2 pathogens. So it probably doesn't have to do with the vector itself or the reservoir host in nature, but instead more of the environmental factors. So for example, in the upper Midwest, we have colder winters and hotter summers. The extremes of temperature are the greatest than they are in other parts of the country, just how cold it can get up to how hot it can get. And because of that, that influences feeding behavior of ticks, and it influences various nesting behavior of some of the reservoir hosts. And so some of that, without getting into all the details, is probably what's driving. It's creating factors that are favorable for transmitting these new pathogens that you're not seeing in the Northeast. So for example, because there's a shorter season between where it gets too cold or too hot, you have the nymphs and the larvae that are more likely to be feeding on the same animal at the same time. So they're more likely to transmit the same pathogen from one to another with only like a transient infection of the host.
One question before we probably end. I think that I totally agree with you that the viral portion of this is really like the untapped, unexplored area.
Mm-hmm.
We see though that when we see patients clinically, they're like a little, a week or more into their disease by the time they present to the hospital. And that one of the big challenges is what sample do we test and how do we test it to catch those viruses that maybe this is more like of an immune mediated thing that we're seeing now and not so much with the virus present. What's the biggest challenge in approaching the viruses, and how would you sort of see the testing going?
Yeah, that's a great question, Kyle. So we just finished validating our Powassan virus PCR. One of the challenges is it's an RNA virus, so you have to worry about RNases in the samples and stability, although we were thankfully able to show a 14-day stability. But that's one of the challenges you have to overcome. And of course, if you're designing an RNA, or an RNA virus detection assay, you have to deal with reverse transcriptase and et cetera. But regardless, what I think once we start using our Powassan virus PCR is I think we're going to start detecting more cases that we didn't even know were out there. If you look at the CDC reports of Powassan virus, I'm just using that as an example, but you only get the cases of reported disease where it's neuroinvasive. There's probably 10, 20, 50 times that number of infections that are mild and not getting picked up. So I think we're actually going to find that there are a lot more viral infections than we think.
Great. Dr. Pritt, thank you so much for joining our 794th monthly lecture series. It was a fantastic lecture, and we really appreciate you taking the time, especially since it's so late in Portugal and there's so much to do at ECMID. So we really appreciate it.
It was my pleasure. Thank you all. Have a great night. Kyle, thank you again for the invitation.
Yeah, we'll be in touch. I hope to see you at